How does RFLP work in forensic DNA analysis? Imagine trying to solve a mystery with a puzzle made of tiny pieces. Each piece is a fragment of DNA, and how they fit together can tell us a lot about who they belong to. That's what restriction fragment length polymorphism, or RFLP, does in forensic science. It's a method that helps identify individuals by looking at differences in their DNA. The process starts with collecting DNA from evidence like blood, hair, or tissue found at a crime scene. Once the DNA is isolated, scientists use special molecules called restriction enzymes. Think of these enzymes as tiny scissors that cut DNA at specific sequences, usually 6 base pairs long. These sequences are like unique markers in the DNA code. If a mutation or variation occurs in these sequences, the restriction enzyme might cut at different spots or not cut at all. After the DNA is cut, it turns into many fragments of varying lengths. To see these fragments, scientists use a technique called gel electrophoresis. They place the DNA fragments in a gel and run an electric current through it. Smaller fragments move faster and travel farther, creating a pattern of bands. To make sense of this pattern, a labeled DNA probe is used. This probe binds to specific DNA regions, highlighting certain fragments and making the pattern clearer. In forensic cases, each person's DNA produces a unique pattern of bands because of the natural variations in their genetic code. These patterns are called DNA fingerprints. When investigators compare the pattern from the crime scene with a suspect's DNA, they look for matches. If the patterns align, it strongly suggests the DNA came from that person. If they don't match, the suspect can be excluded. RFLP was one of the first methods used in forensic DNA analysis because it's highly specific. It also shows 2 different fragments at a single location because humans have 2 copies of each chromosome. This increases the accuracy of identification. However, RFLP needs a lot of high-quality DNA and takes a long time to process. It was used in many early criminal cases, paternity tests, and genetic studies. In the end, RFLP works by exploiting tiny differences in DNA sequences that affect where restriction enzymes cut. These differences produce unique fragment patterns that can be matched to individuals. This method helped shape the field of forensic science and set the stage for more modern techniques used today.